Ever since we learned about the new coronavirus that causes COVID-19 late last year, researchers and scientists around the world have been scrambling to find a way to stop it. As of today, there are almost 350,000 confirmed cases of COVID-19 worldwide, and more than 15,000 people have died. Governments are asking people to practice social distancing to slow down the spread and experimenting with different treatment methods. But the one thing that would help get this thing under control, and which is still missing, is a vaccine. The race to develop one and get it to the public as quickly as possible is on, and Canada is part of that effort too.
Today, I can announce that we're providing $192 million to directly support vaccine development and production in Canada.
Today, we're talking to a couple of people on the front lines of this race to get a COVID-19 vaccine ready for public use. A volunteer in one of the first human vaccine trials in the US, and a professor at Western University who's part of a team that's working on a vaccine here in Ontario. From Global News, I'm Tamara Kandaker, and you're listening to Wait, There's More. Neil, can you just tell me a bit about yourself, a bit about your background to start off?
Sure. I was born in Alabama, grew up most of my young adult life in Georgia, and moved out to the Seattle area in '97 to start finding jobs in the IT market. I'm a network engineer for Microsoft. I lived out here in the same area for the last 23 years now.
And do you have a family?
Yes, I have a fiance who has a daughter, and I have two of my own. And together we have a nice little blended family with 4 young ladies.
And so you live in the Washington State area, and Washington State's been one of the hardest hit places in North America with this coronavirus.
We represent about 2% of the population of the United States, but we represent over 20% of the infections. Today it's 796 infections. Never since World War II have we faced a situation like this where we both can be part of this battle, but must be part of this battle.
It's definitely been hard hit. I actually live approximately 5 miles from where the outbreak happened at that retirement assisted living home. We've been going through almost every other day another series of lockdowns. We're expecting any day to have a shelter-in-place order, much like you're seeing and hearing about in the San Francisco area.
There's a mutuality and there's a recognition of our interdependence that requires of this moment that we direct a statewide order for people to stay at home.
Social distancing has become the new norm for us.
And this vaccine trial, can you tell me a bit about it? How did you find out about it?
So I found out about the trial through Facebook, through a friend, and I figured, it sounds interesting. I'll do a little research about it. It's very new. Not a lot has been done in this area, but it's safer in that there is no dead or weakened coronavirus in this at all. So the dangers are very limited in terms of that. I can't catch the coronavirus from the vaccine. Without getting too deep into the weeds, it uses messenger RNA that's injected into my body, invades my cells, and teaches my cells how to make the proteins that I'm sure you've seen hundreds of pictures of what the coronavirus looks like, the little sphere with small triangles poking out. So my body is building proteins that look just like the outside of the coronavirus in order to trigger my white blood cells to learn how to attack and kill that type of protein on the outside, so that should I become infected, ideally, it already knows how to find it and react rapidly and can kill the coronavirus before it can take a hold and cause me physical harm.
That's really interesting. So because you're not being injected with this virus, does that explain then why this trial is happening so quickly? Like, I understand that animal trials were just skipped altogether, which is usually one of the stages in vaccine trials, right?
That's right. Yeah, I and the 44 other people taking part in this are the animal trials in this instance. Part of it is because it doesn't have the virus in it, but another part is because of the need of the world to have something to fast track in order to get this under control.
And why did you decide to volunteer to do this?
I really do feel like it's all of our parts to play in helping humanity. I mean, we're all part of the same race. We all live on the same planet. And, when I have to tell my kids it's time for your shots, go get your flu shots, get your vaccines. I mean, what better example than Dad's helping too? He's getting his done. I want them to grow up in a world where hopefully we don't have things like this happen in the future. So if I'm healthy and I can contribute a little bit to science along with the other 44 people and the amazing staff at Kaiser Permanente Research who are doing this, then why wouldn't I?
We just talked about the fact that you're not going to be injected with the virus, the danger is minimal, but what were you told about the risks that you might be taking on by participating in this trial? What kinds of conversations did you have with your doctor before you started this?
So they were very thorough in going through my medical history to look for underlying conditions that could be complications, going through any surgeries I'd had in the past, making sure that I had good heart rate, good pulse, blood pressure was okay. They drew several vials of blood, sent those off to the lab to look at other enzymes in my blood and make sure I was healthy. Obviously, this is an unknown. It hasn't been done before, so they were worried. They said that there could be anything from mild soreness at the injection spot to tightness of joints, swelling of joints. There could even be headaches, nausea. You could even develop a runny nose and a fever. So they were pretty clear about what the risks were. They seemed minimal. I knew I couldn't catch it, so I wasn't worried from that aspect. And they did send me home with a daily log where I record any symptoms whatsoever. They have me record my temperature daily. And I go back once a week where I was injected with a vaccine so that they can do a blood draw and analyze how my body's reacting to the injection.
And what are they trying to find with these blood draws? What's the goal of this particular trial?
So because they know that my body should be producing proteins that look like the coronavirus's exterior, they're looking that, number one, that is happening. Number two, that my immune system sees it as a threat and is reacting to it. And number three, is successful in building the antibodies to fight off and kill those little fake coronavirus proteins in my blood. After I go for 4 weeks, each week with another blood draw, on the fourth week, they will give me another vaccine injection. And then I'll go through another 4 weeks of blood draws. And that second injection is to simulate more of an actual infection to gauge how quickly my body responds to another invasion and shows that hopefully it responds quickly and it's fighting it off successfully.
And your family, how have they reacted to your decision to do this?
So my fiance is a registered nurse, so she understands medicine fairly well, and she is very supportive. She thinks it's great that I'm trying to help out. My girls all think it's amazing. They're a little bit more starstruck with the whole dad's on TV rather than dad's actually doing something good for the world. But I think as they get older and they look back, they'll figure out pretty quickly that there was more to it than just being on the news.
Well, I think I speak on behalf of a lot of people when I say thank you for doing this, and I really appreciate it.
Thank you so much. I appreciate the staff at Kaiser Permanente Research. They are honestly working 18, 20-hour days and even weekends. They are really an attentive staff, and the other 44 volunteers are just as important.
Okay, Neil, thank you so much.
Okay, thanks so much, guys.
We'll be right back. So the trial that Neil is part of is one of many that are going to be taking place over the next little while. Companies and universities all over the world are trying as fast as they can to develop a vaccine that's safe and effective. And we wanted to talk to someone who's part of that effort. Chilean Kang is a professor of microbiology and immunology at Western University in London, Ontario. He's worked on creating vaccines for HIV and the Middle East Respiratory Syndrome, also known as MERS, which is another coronavirus that's closely related to COVID-19. And he's now part of a research team that just started developing a COVID-19 vaccine. Hi, Professor. Thank you so much for doing this.
It's my pleasure.
So earlier we spoke to a man named Neil Browning, and he's a volunteer in the human trials that have already started for a vaccine candidate created by this Boston-based biotech company called Moderna. So that is one of 35 companies and academic institutions that are now racing to create a vaccine.
If these drug tests are successful, then we will have some tools in our toolkit to help people keep from getting infected. But, the holy grail here is a vaccine.
And I know at least 4 of them are doing animal testing. We have this one that's already doing human trials. And I understand it usually takes a long time to even get to this stage. So how have we gotten here so fast with the coronavirus?
I think this company was planning to design a vaccine against the MERS coronavirus and the SARS coronavirus prior to this COVID-19 pandemic. And they had this technology established. And so they are trying to adopt this technology to make a vaccine against the COVID-19.
Right. So they're building on that work. But also part of the reason for why we were able to get here so fast is that China released the sequence of the genetic material of the virus, right? How did that contribute to this process being accelerated?
Yes. Yeah, at the end of 2019, China isolated the virus and sequenced them, and so that they know what the sequence is, meaning that it is possible to construct those sequences or synthesize even those sequences and make proteins on the basis of those sequences. And they found that the infecting virus to cause COVID-19 is known as SARS-CoV-2. So SARS-CoV-2 viral genome has been sequenced in China and released, and that information was useful to pick specific sequences out of that to make vaccines. And so that information was available in the early January, and they used this sequence to make their vaccine.
Right. So China shared this information in January, and then research groups around the world were able to use it to start growing the virus and studying how it sort of invades the human cells and makes people sick.
Yes. Well, that sequence is very useful to design a vaccine because you don't want to use entire virus genome, and you only need the genome which can produce those spike proteins.
Like other coronaviruses, the particles of the virus that cause COVID-19 are sphere-shaped proteins, and they're covered in spikes. Those spikes lock onto receptors that are on the surface of cells that line the human lungs, and that's how the virus breaks into those cells and infects them. And once they get in, they hijack the cell's reproductive machinery so it produces copies of itself. And like Neil mentioned, in the clinical trial he's part of, they're trying to get the body to make proteins that look like these spiked proteins.
There are 2 other proteins on the surface of the virus. Is known as envelope protein. The other one is matrix protein. So the E, M, and S, those are 3 proteins on the surface of the coronavirus. And if you make a vaccine on the basis of those 3 proteins, you may be able to prevent infection of coronavirus.
We're already building on the work that was done to develop vaccines for SARS and MERS, and I understand that you've also worked on a vaccine for MERS. And I'm curious, how far did we get with developing a vaccine for those 2 viruses? With SARS, there was never a vaccine that was fully developed, right?
Yes, because of the demands for the vaccine against the SARS, actually interest has reduced because the SARS was contained after some 8,000 infections around the world, but that was contained. Therefore, not so many people were interested in developing or further developing the vaccine against the SARS. The MERS coronavirus, on the other hand, had about 2,500 infections with a very high mortality rate, and that had about 36% mortality rate.
At least 30 people worldwide have died from the infection called MERS for Middle East Respiratory Syndrome. Dozens more have become sick. Experts from the World Health Organization say the virus is a threat to the entire globe.
And people were interested in making vaccine that too was contained. So not so many people are interested in making vaccine against the MERS coronavirus. But there are certain companies who are willing to develop vaccine against the MERS coronavirus just in case it reemerges in the future.
Generally, once an outbreak subsides or once an outbreak is contained, I guess the interest in funding a vaccine, in funding vaccine research, it goes down.
That's correct. Actually, government grant continues up to a certain level, but the industry and the pharmaceutical companies may lose their interests if there's no more problem with a certain viral diseases.
Okay, and the work that was already done to develop a vaccine for SARS and MERS, how helpful has that been to develop a vaccine for the virus that causes COVID-19?
It is very important to have that experience because we know how to construct the virus and which gene that we can use to have efficient vaccine development. And the experience that we have with the MERS coronavirus really helps us to design the SARS or COVID-19 vaccine. So we have started our research to develop vaccine against COVID-19 on the basis of our experience with the MERS coronavirus.
So there are 3 different types of commercially available vaccines that Professor Kang told me about, and this next section is a bit technical, so bear with me. The first type of vaccine is a live vaccine, which uses a weakened form of the live virus.
You're using live virus as a vaccine, and our body can react to it and make protective immune responses. And these include live poliovirus, measles, mumps, rubella, and so on.
The second type of vaccine is the inactivated vaccine, which injects a killed version of the virus. So the virus's ability to replicate is gone, but it's still intact and the immune system can recognize it.
You can propagate the virus and purify it and then kill them, inactivate them, and use that as source of antigen to trigger immune responses.
Another type of vaccine he describes is the recombinant virus vaccine, which injects specific pieces of the virus, like its protein, into the body. That triggers a response from our immune system that targets that specific piece of the virus.
Those work to generate the neutralizing antibodies, and these antibodies can protect the infection of the live virus. And those are 3 different commercially available virus vaccines. But now people are using mRNA vaccines, DNA vaccines, and so on. There are many other strategies that are being used to develop the different types of virus vaccines.
And the one in the clinical trial we talked about at the beginning is an mRNA vaccine, which doesn't infect you with the virus at all.
So mRNA vaccine is none of those 3. It's a new idea to produce proteins on the basis of one type of nucleic acid called ribonucleic acid, and the messenger RNA or mRNA can make a protein when it gets into our body.
A protein that looks like the spiked protein of the coronavirus.
And that protein then triggers immune responses against the infecting virus.
In order for a vaccine to work, it needs to be able to get our immune system to produce antibodies in the blood, which would recognize and remember and defend against the virus if we get exposed to it.
That's correct.
And so traditionally, to get the body to produce antibodies, vaccines will inject people with the virus, like we just talked about, maybe a live weakened version or an inactivated version of it. And what are the drawbacks to doing it that way?
The live virus vaccines have a tendency of converting back to the wild type virus so it can cause a disease. For example, if one uses oral polio, which is a live poliovirus vaccine, very few people or children who are vaccinated with a live poliovirus vaccine, they convert back to the wild type and create paralytic polio.
So what Professor Kang is saying is that the injected live virus, even after it's been weakened, could potentially turn back into its pathogenic form and make the host sick, although that's very rare.
So that's one of the problems. And the killed whole virus vaccine doesn't have much of the problem, but it requires additional substance called adjuvant to stimulate the immune responses, so that sometimes one can have the adverse reaction to the other substance mixed with the vaccines.
The other potential issue with the killed whole cell vaccine, according to experts, is that they may not always produce an immune response, and sometimes you need several doses for the body to react.
The third type, that the recombinant protein vaccine, is so far is working very well, and it has minimum amount of the side effects, and it triggers immune responses. However, there are only two different kinds of vaccines available using this strategy. So scientists have to develop further how to make vaccines effective to prevent different virus infections.
Okay, and so the mRNA vaccine that we talked about, is something like that lower risk? Is it faster to produce?
Well, there are advantages is that it may have very little side effects, but there are some challenges with mRNA approach. First of all, production of mRNA in a large quantities is difficult. Secondly, mRNA is highly unstable, so when you inject into your system, that may degrade very fast. And also, the amount of the mRNA injection could be limited. In other words, you can only inject a certain amount. I mean, this trial, they inject anywhere between 20 microgram to 250 micrograms. Okay, so if you want to inject 250 micrograms as effective vaccine, the supply of this vaccine could be very limited. And then also, we don't have enough evidence to demonstrate that efficacy of this mRNA approach. And so you have to make large quantities of proteins after injection of mRNA, but it is not clear how much protein you can generate from these mRNAs. So there are several challenges with this mRNA approach.
The process to make a vaccine available to the public is quite long.
So we need to make sure it's safe, and we need to make sure it works. That entire process will take at least a year and a half.
Can you just kind of take us through the steps that a vaccine candidate has to go through before it can reach the public?
Yes. It's a long process. No matter which strategy you use, the developed vaccine has to be tested in animals. Once it works in animals, and make sure that it is safe to use in humans, then you go through the different stages of the trial. And there are 3 main trials one has to go through. The first one is to see whether or not the vaccine is safe. The second trial is, we call it phase 2 clinical trial, is to look at the immune responses in the vaccinated people. And the third trial is the efficacy trial to see whether or not the vaccine works. The vaccinated group should be completely protected compared to the non-vaccinated or control groups.
Right.
So that's how you demonstrate the efficacy of the vaccines.
So that first stage is usually with a few dozen volunteers to monitor sort of side effects, right? And then...
That's correct. The second phase is to see whether these vaccinated people will produce the adequate neutralizing antibodies in their body. And then the third trial is to vaccinate a large number of people or volunteers to see whether or not the vaccine really prevents infection. Then you apply for the license, and the licensed vaccine then can be marketed and distributed. Now, in this COVID-19 case, since we are in an urgent demand of the vaccines, people bypass some of the steps.
So essentially, we have to confirm through all these different stages that the vaccine is both safe and that it actually works.
Absolutely.
And what are the risks that come with rushing this process?
Well, since we are not going through the preclinical trials, some of the vaccines may cause undesirable side effects. And for example, entire spike protein may have some toxicity in it, and so that it can be toxic for the recipients of the vaccines. However, sometimes the people go ahead and vaccinate people if the benefit is much greater than the risk that they take. So this particular vaccine, mRNA vaccine, we'll see whether or not these people can induce desirable immune responses. And if so, then they may be protected from natural viral infections.
I know there are also treatments for COVID-19 being tested around the world while a vaccine is being developed.
And so we have a trial going at the moment to see if we can prevent you from getting infected with the virus. That's with a drug called hydroxychloroquine.
Can you tell me anything about those?
People are using many different types of antiviral therapeutic agents to treat the COVID-19 patients. However, virus reproduces only in the living cells. And if you try to knock out the virus function in infected cells, you damage the host cell as well, so that you have tremendous side effects. So it's very difficult to find effective antiviral therapeutic agents. Because of the urgency of this disease, people are using so many different therapeutic agents. Mr. President Trump mentioned about the malaria drug.
I have a feeling you may, and I'm not being overly optimistic or pessimistic, I sure as hell think we ought to give it a try.
Yeah.
And some are using the retroviral therapeutic agent in India, and some are using the drugs which can treat influenza virus infections and so on. But all these antiviral therapeutic agents may not work effectively because we have a problem with the side effects. And also there's not many viral-specific antiviral reagents are available.
So overall, then, it sounds like a vaccine is our best possible hope to fight this thing.
The vaccine is the best and the most effective way of preventing this crisis.
There are certain people who are immune to the virus, right?
Since there are so many different coronaviruses in nature, maybe some coronaviruses may cross-protect itself. For example, SARS coronavirus uses same receptor on cell surface of the human cells as the SARS-CoV-2 or the virus which causes the COVID-19. Okay, so if someone is immune to the SARS virus, then that person may be cross-protective against this SARS-CoV-2. So there might be some immunity built by unknown infection previously.
And so, Professor Kang, last question. Given that the vaccine could take a really long time to develop and reach the wider public, what is our best defense right now to containing this virus?
Well, the government is advising everybody to have a social distancing, which is important, so that you don't interact with people as much as you used to. So the 2-meter social distancing is highly recommended, and the people wash their hands frequently because if you have a contaminated hand and touch your face, you may have a better chance to transmit the disease. So washing hands is very important.
Thank you so much, Professor, for explaining all of this to us. We really appreciate it.
It's my pleasure.
All right, that's the show. Thanks so much for listening. But before we go, just a few updates on how provinces are ramping up their social distancing measures even further to stop the spread of COVID-19. Today, the premiers of Quebec and Ontario ordered all non-essential businesses to close down.
The next 36 hours will give non-essential businesses the chance to adapt and prepare. Tomorrow, we will release a list of businesses that are permitted to stay open.
Also, Vancouver's City Council voted in favor of imposing $1,000 fines on people who violate the city's social distancing rules and $50,000 fines on businesses who break the rules.
If residents do not immediately change their behavior and begin acting with their fellow Vancouverites in mind, I will work with local retailers and I will take action.
That's all for now. I'm Tamara Kendacker, and I'll talk to you tomorrow.